All right, let's get started. I graded the homeworks. I think about 2/3 of you lost 2 points because I really wanted you to use variables in the indices for figuring out, like, where is the top box, what is upstream of it, what is downstream of it. Because if you changed the sequences at the top, it would be nice if the rest of the code worked. Right? Like that's like the point of code. And I feel like I did not make that particularly clear because about 2/3 of the class did not do that. About 1/3 of the class did. I'm wondering how strongly that's correlated to people who've had programming before, probably pretty strongly. So I'll try to think of a way we could do some makeup points for that. How about this? If you guys can rewrite the code and show me using variables instead of using like the number 56, the number 57, and send it back to me, I'll give you your points back. But I just want you to like wrap your head around like that's the point of like writing your Python code, is that like you could reuse it again and again and it would keep working. Like you could have thousands of bases and have no idea where the start codon is, but you could reliably use that code to find the start codon and like get all the sequences after it. So send me back another version with your variable driven indices and I will give you those 2 points back. I think that's a fair thing to do. But other than that, and the homework today, we're done with Python. Say after Jay if you want help with the Python homework. If it makes my life easier if you send it to me in a format I can just run it. I can type Python, Daniel's homework, whatever the file is, and it just runs. So if you can do that for me, that's awesome. And there are a couple of you that the plain text you sent me was not plain text, and I think I sent you a little comment with your homework, like please try to figure out like how to make things plain text. But for the most part, it worked for most students. So yeah, we'll work on that after class today. So yes, Monday, November 5th, project proposal due. Don't forget about that. And let me know if you have any questions. So today I'm going to be doing a lecture, kind of getting us oriented around RNA-seq, and then Narula is going to be doing the lab. So we'll probably have a little break where we swap computers and turn the Zoom on and get all that worked out. Any questions, guys? Nope, that is not what I wanted to do. Nope, yes it was. Okay, so I want to start with a little bit of background. Most of you should probably have had this from biology. Oh, this is not, I want my pointer. Oh. Okay, good enough. So when we're sequencing RNA, there are a couple different ways to target RNA, and I want to cover both because we have people working on a diversity of different systems in this class. The example we'll see today is targeting mRNA with poly-A enrichment. And this, if you're working with an organism that puts poly-A tails on its RNA, this is a very convenient way to pull out the mRNA. And it's the basic idea is use a poly-T oligo that's attached to a magnetic bead or some other kind of pull down procedure and pull down any piece of RNA with a poly-A tail. So of course this is only going to work if you have actual poly-A tails. But this is going to mean that you are sequencing the untranslated regions and the coding regions, but it's a mature RNA. So that means the introns have been spliced out. You're not getting all that flanking genomic DNA. You are getting the untranslated regions that are transcribed, but you're not getting anything else upstream or downstream, and the introns are gone for the most part. Of course, all of these lab procedures are a little bit leaky, so you may get some other RNAs, but for the majority of the time, this is how you would primarily get your mRNA. Yep, so just taking advantage of that poly-A tail. If you are working with a microbial organism that does not have poly-A tails, your primary goal and what you primarily want to get rid of is ribosomal RNA. And ribosomal RNA will be 60 to 80% of your sample if you just sequence total RNA. If that's what you want, that's great. But most people do not want 60 to 80% ribosomal RNA, especially if you're working with a single organism or a single isolate, because you would have like 1,000,000x coverage of that one rRNA and not much coverage at all of your coding regions, which is what you might primarily care about. So in this case, there are ribo0 and the other one is called, there's a number of different kits you can buy that basically are able to subtract out the ribosomal RNA from your RNA pool. And that's a nice way to enrich for mRNA. You're still going to get other non-coding RNAs, but the ribosomal RNA is primarily what's going to cause you the most problems. This is usually done biotinylated capture probes, but I think there's a number of different kits out there that you can try out. You do want to select a kit that's specific to, not to your organism, but to your like realm of life. Like there's a plant kit and like a bacteria kit and a fungal kit. And so pick something that's going to know what your ribosomal RNA looks like. This is cheaper than the poly A selection. So even if you have a eukaryote that has poly A tails, some people do this just because it's a lot of labs do not charge as much for it. For most of kits that create rRNA libraries, they have strand-specific sequencing these days, which is basically you are able to sequence, when you sequence something, if it came from the forward or the reverse strands, it's only going to sequence it in one direction. You get that information back. This is really nice for assembly and for mapping. You can differentiate overlapping genes. So like, let's say you have a segment of the genome and you have one gene that's like pointing this direction and one gene that's pointing this direction. They actually overlap a little bit. If you don't have strand-specific sequencing, it can be hard to sort out if it's just one giant gene. But if you know which strand your reads map to, then you can kind of see that they're two different genes. And that doesn't happen a ton, but it does happen. You can figure out pseudogenes more easily, antisense transcripts, if they are appearing, what's causing them. And so that's actually just a really nice informatics. If you're doing assembly, this makes your assembly a lot easier because you know what direction your reads are going in. So all of that should be available for most sequence providers these days. So that's pretty much all I have on like the beforehand library prep part of RNA-seq. It's good to just visit with whoever's going to be doing your sequencing and learn more about what kits they use and how they work. And there's just a diversity of different stuff out there, and I don't even know probably half of it. So so whatever your lab is using, try to learn about it. And if you have any specific questions, I'm happy to try to answer them. But there's a lot of ways to prepare RNA-seq libraries. So let's talk a little bit about what are your experimental goals if you're doing specifically mRNA-seq. Sometimes you're just looking to do a catalog of genes. So maybe you have an organism and there's no genome and there's really no genomic information, and you're just trying to get a toehold into how do I start doing molecular research here. And the first thing you might do is just take one isolate or one individual and sequence RNA and just get a catalog of what genes do I have in this organism. From there, the next question, because RNA-seq data is quantitative, you can count the number of reads per genes. You can look at the expression level of those genes. And once you know expression levels and you have quantitative numbers, you can do statistics, and then you have statistically significant differentially expressed genes. So at this point, you might say, okay, I have a plant, I'm going to expose it to drought and control treatments. And then I will sequence the genes and see how expression has differed. The great thing, and this all used to be done with microarrays, little chip-based gene probe systems, and largely the world has moved on to RNA-seq because you get a lot more information. You can actually ask the same questions like, what do I have and how highly is it expressed for individual alleles and for splice variants? Because that RNA sequencing data is actually going to reveal the differences in your two alleles, if there are sequence differences, and how the splicing actually happened. You don't get that from a microarray chip. Another reason to do this is to annotate a reference genome. So if you have assembled a reference genome and you don't know where the coding regions are, you can sequence a bunch of RNA and map it to your genome and say, oh, look, that's a gene. I can tell because it's transcribed. So that's an important reason to do this. Variant genetic marker discovery. I work with some people who have conifers as a primary interest. They have 30 gigabase genomes. Nobody wants to sequence the DNA of a 30 gigabase genome. It's not productive. So they do a lot of their variant calling only from the genes, only from the transcribed regions. So you can do that with RNA-seq. Another way to do that is to do exome capture. But in some systems, it does make sense to do your variant discovery. Like we learned with DNA, you can do some of that also with RNA. And finally, RNA-seq is also great for looking at post-transcriptional modifications and RNA editing, because you actually get the sequences. So if you compare the DNA, compare the RNA, and see what happened post-transcriptionally. So that can be quite an interesting project. Here's an example of what the genome annotation would look like. You map your, and this is good, like you should look at your RNA data. And we're gonna talk about that because when we were looking at SNPs, we pulled it up in IGV, we pulled it up in SAMTools TView, and we looked at what do our reads look like. Can we, this crazy software tool I just ran told me that there's a SNP at base 168. If I go to base 168, does it look like there's a SNP there? Does this seem like it's real? You wanna do the same type of thing with your RNA data. But if you're mapping your RNA data back to an organism, back to a reference genome, you're gonna see a very different structure. So you're gonna see it piling up where your exons are. So you shouldn't have many, if any, reads mapping to your introns, like in between, but you'll see lots of reads piling up at your exons. And your reads would often be split. So because this splicing has gone on before you sequence it, a read will start in this red region, and then the rest of the read will be in this red region. So when you map back to the genome, there's these big gaps. So this is kind of a typical what it looks like when you map RNA back to the genome and visualize it. Since it's so easy to produce very, very, very deep RNA sequencing these days, we often visualize it, instead of individual bases, you might want to look at it as more just a density plot, kind of like this, where you can see stacks of how many reads have aligned. But if you use something like IGV, you can zoom all the way down to the base pair level. So yeah, it's important to think about alternative splicing. So just a reminder, for those of you who haven't had a molecular class in a while, in certain organisms that have exons, your exons can be differentially arranged to make new and interesting transcripts. So you have a single gene. This one, in this case, has five exons, but sometimes they're spliced together like this, sometimes they're spliced together like this, and sometimes they're spliced together like this. So we get all of these different interesting isoforms of our gene. And the great thing about sequencing is if you sequence across these transitional points, you can figure out what isoforms you have, what alternative splicing you have. And that looks kind of like this. It's an interesting thing to think about, and kind of similar to some of the things we talked about with assembly. You don't always have enough information to, like, let's say you have a gene and these are your two different isoforms. Sometimes you have this exon in the middle and sometimes you don't. If you have reads that map solely to this exon, that's not going to tell you which alternative splice form you have, because this particular part of the gene is present in both isoforms. If you have reads that map into this exon, that is very informative. You have to know that you have an isoform that one that has this exon and one that doesn't. And then if you see it in your reads, you know that that's what you've sequenced. And then again, over here, if you have reads in this area, it would not be informative. This is where paired-in reads are again useful. We talked about how paired-in reads were useful for DNA. This is another way that they're really useful, particularly for RNA. For example, here, one of our reads maps into an uninformative region, but the other one maps into a very informative region. And this one, a read maps into an uninformative region. And what we would think is maybe an uninformative region, but since we know our library size, and we know that it's very unlikely we would have selected and sequenced a fragment long enough to include this entire exon in between your two reads, you can deduce that you have isoform B. Does that make sense? Because you know how far apart those reads are, so you can say, oh, look, that's definitely isoform B, because there's not enough room in between my reads for that exon to be in there. So that's how you use your paired-in sequencing data. And you have to have a well annotated genome where you understand all the isoforms available, which when you work in trees like I do, that's never the case. But some of you work in fun organisms where there's lots of knowledge and you might actually have that kind of data. I will have this slide both today and on Monday. This is the hardest thing, I think, for people to sometimes wrap their head around, and I get lots of questions about this. RNA-seq gives you relative abundance only. So a good example, Halloween's coming up. Let's say I have a huge bag of M&Ms, and it's completely homogenized, like it has been mixed thoroughly. And I give you a sample of 100 M&Ms and say, tell me the ratio of red to green to brown. You could tell me, oh, you got a crazy lot from the factory. It is 80% red M&Ms. And you would know that because you took a sample of it. If I asked you how many M&Ms are in the bag, your little cup sample does not tell you that. If I have a big bag of M&Ms and a little bag of M&Ms, you can tell me this bag has a higher ratio of red to brown than this bag. You cannot tell me from that sample which bag is bigger overall, right? So RNA, we'll talk a lot about statistics on Monday, but I want you to start thinking about it. It's a sampling. You're sampling from a larger pool. You're not quantifying absolute abundance, only relative abundance of these genes against each other. Other limitations of RNA-seq, reverse transcription, PCR fragmentation, all of these steps can introduce biases. If you have really high quality RNA and lots of it, you can go to a PCR-free prep, and that will reduce your bias significantly. But not everyone has the luxury of doing that. But it's worth noting that you can see depletion of reads at both the 5' and 3' end, which means if you have short reads, you can have problems sampling them adequately. You may underestimate the expression level of short genes. You can also get GC bias, and you can get length bias and how these are amplified. So just things to be aware of. If you can do a PCR-free prep, that's preferable. The next thing I want to talk about is project design. If you guys have any questions as I kind of fly through these, feel free to shoot your hands up. We can stop. Okay, so the 3 important things for project design, in my mind, are read count, replicates, and cost. I can't really help you with cost. You're on your own on that one. If you come talk to me, I can tell you companies that we have worked with that we've liked. I can tell you companies we have worked with that we don't like. I can tell you who's running a good deal right now, at least what I know of. You can sign up for all of the Novogene emails, maybe get a discount, but for the most part, costs, you're on your own. But I can tell you a lot about how to design your project based on read count and replicates. My other advice, and I feel very strongly about this, if you're an ag campus student, we have a statistician in the animal science department, Liesel Schneider. She is available for consulting. If you are on the other side of campus, we have a whole consulting group run by Bob Minchin. He and his group are also available for consulting. Please consult somebody before you design some crazy big experiment and then run it, and then your stats fall apart and you can't do statistics. Probably most of you have had a basic statistics course. You've learned about basic experimental design. You've learned about factors. You've learned about blocking. If you need to revisit that, we have statisticians on campus who are there to help you. And just, it's really hard to find out that you just spent $20,000 on something that you can't analyze. So don't do it. Nobody wants to hear that. And your resident statistician and/or bioinformatician probably likes coffee, so buy them a cup of coffee while you're at it. It's my recommendation. Yes, and if you have any questions, you can come talk to me. You can come talk to Liesel. You can email Bob. I can give you any of those email addresses. So moving on from there, let's start with replicates. We'll also talk about read count. We're going to start with replicates. I apparently have a lot of extra slides there. Okay, so most of you have probably heard about biological replicates, but it's really important to revisit biological replicates in terms of RNA-seq. So biological replicates are independent biological samples processed separately, and in the case of sequencing, you want to barcode those libraries separately. Technical replicates are when you're maybe doing the exact same biological sample, but independent library construction, or maybe sequencing it, the same biological sample on like 2 instrument runs. So that's where you're trying to measure technical variation that's being induced in your experiment, versus biological replicates is when you're trying to actually measure the biological variability of the phenomenon that you're studying. Technical reproducibility is very, very good for RNA-seq. We have tons of citations for this. An individual sample, if you prepare the library multiple times and you sequence it on multiple different instruments, you will largely get the same answer. Biological variation is very, very big. Gene expression is extraordinarily variable. So when you're doing RNA-seq, you want to put all of your time and effort and money into biological replicates, because that is the vast majority of your variation, and that's what you need to capture. There's not, as long as you're doing your technical reps, as long as you're doing your technical aspects consistently, like you're using the same library kit and you're using the same RNA prep and you're using the same instrument, the technical variability should be just almost nothing compared to your biological variability. So twice a week, I get asked, how many replicates do I need to publish? What's the bare minimum? And I'm going to tell you now, it is almost impossible to publish with 1 rep, as it should be. There's a reason why it's almost impossible to publish with 1 rep, because it's just not very, it's not very good data. Like, it's just not very statistically compelling. I still see tons of publications coming out with 3 replicates. So I'm going to tell you that that's your minimum to get published. And I'm going to give you a whole bunch of great papers. If you're in the RNA-seq, read all these papers. They're wonderful. But this one says specifically, beyond a depth of 10 million reads, replicates provide more statistical power than depth for detecting differential gene expression. So once you can afford up to 10 million reads per sample, add replicates. That's the formula. Some people are going to argue with me, and I think I have this on some additional slides, that it should be more like 20 or 30 million reads. But somewhere in that 20 to 30 million read region per sample is when you want to tip over into like, okay, more biological replicates is going to give me more statistical power. This was a really cool paper that came out that had 48 replicates. They decided to just go the distance, specifically to study the statistics of RNA-seq. This was in yeast, I believe. How variable yeast is compared to your system is up in the air, but it is a toehold into the statistics here. And they had some interesting conclusions. With 3 biological replicates, 9 of the 11 tools evaluated found only 20 to 40% of the significantly differentially expressed genes identified with the full set of 42 clean replicates. So what this means, if you had, they're making the assumption that 42 replicates is enough, that it was extremely, extremely accurate. Looking back, if they had only sequenced 3 replicates, they would have only identified 20 to 40% of the actual biological variation. So while I'm telling you that 3 replicates is publishable, I am also telling you that 3 replicates means you are going to miss more than half of what is actually biologically going on. These results suggest that at least 6 biological replicates should be used, rising to at least 12 when it is important to identify significantly differentially expressed genes for all fold changes. So if you're looking at a gene that might have some really subtle differences, 12 biological replicates is where you want to be at. Nobody wants to hear that because it's very unaffordable. But six is a good metric. We're still kind of stuck with some projects where we can't afford more than 3, and it's very depressing. But the projects where I do go up to 6 if I possibly can afford it. So that's what I recommend. And as far as tools, the other interesting thing about this paper that's worth reading, if fewer than 12 replicates are used, a superior combination of true positive and false positive performance makes EdgeR and DE-seq2 the leading tools. So if you go above 12 replicates, there are some more straightforward statistical packages, but EdgeR and DE-seq2 do some kind of statistical massaging to figure out variability across all of your treatments and then apply that. I'll talk more about that on Monday, but those are the tools that I've consistently seen in the literature is really good, especially for these somewhat low replicate number experiments. Just to continue to drive home this point, this is a great paper, RNA-seq differential expression studies, more sequence or more replication. Okay, so let's see. For this graph, we have the number of reads, and they did kind of a random sampling, like back sampling, like what if they sequenced up to 30 million, but then they went back and took subsamples at 5, 10, 15, 20, 25 million. And then they looked at the number of differentially expressed genes, so they were able to detect with each of these series of replicates. And I think this is so funny. Like, this is someone who's so determined not to pay color page charges that we have these like really thick lines, just like determined not to pay that extra $100. But in 2 biological replicates, even if you vastly increase the number of reads, you're not getting as many differentially expressed genes as you do with 6 or 7 replicates and only like 3 million reads. So what this is telling you, your differentially expressed genes, you get significantly more power to detect them as you increase with replicates. And as you add depth, you're not getting much additional power. And over here, this is the actual power calculation, the power at FDR 0.05. And again, at 2, you have almost no power. And as soon as you go up to, in this case, 7, they're saying you have almost perfect power. So, you know, just another thing that's kind of indicating that biological reps are where you want to go. So which to use of EdgeR or DSeq? This is from the S Church paper, which is the 48 yeast samples. For experiments with less than 12 replicates per condition, use EdgeR or DESeq2. For experiments with greater than 12 replicates per condition, use DESeq. I think that's reasonably good advice. From our lab, and Narula can talk to you about this as well, if you have no biological replicates, DESeq2 probably won't give you any results. It'll just say nothing significant. I'm sorry, I can't deal with this. But EdgeR will actually maybe give you some differentially expressed genes. We have often in the past just used both and looked at the overlapping gene set, because they do have somewhat different statistical models for variation. And we kind of want to see like what genes are robust enough to come through both models and still be significant. To save money on library prep, because you do pay per library, that's the vast majority of your RNA-seq costs. People like to pool samples. And does pooling my samples count as biological replicates? I'm going to tell you no. With pooling, you'll get an accurate mean, but not an accurate measure of variability. And variability is what's the big deal in RNA-seq. The literature is very mixed on this issue. Tons of people get away with pooling and publishing pooling. But it doesn't make any solid statistical sense. Go ask any statistician whether or not they think this is a good idea, and they're going to tell you no. You are not modeling a distribution here. You only have the mean. You're ruining your stats. And yeah, and there was a paper, the DEGs identified in pooled samples suffered low positive predictive values. So there are a few papers out there that have tried to examine this. And can you get away with publishing pooled experiments? Yes. But do you want to base your conclusions and the potential future science that you're going to do on data that's not very good? And my argument is going to be no, you probably don't want to do that. So if you try not to pool. Some people, I've also worked with students who have like, I'm working on an insect that's this big. Okay, well, you're going to have to pool because you're not going to be able to get enough RNA out of the insect that's this big to, you know, like sometimes you can't get away from it. But if you can, it's better to individually barcode all your samples. Everyone has probably had blocking in experimental design, but let's revisit it because there's some specific ways that you might want to block in an RNA-seq experiment that are not obvious. So randomized block design is to divide your samples into blocks in order to control for variation between blocks. And you want to randomize the assignment of individuals to treatments in an experiment. So this is pretty basic stuff. But one place that people don't really think about blocking is these lane effects. So lane effects are basically, you can get systematically bad sequencing cycles and errors in certain lanes of a sequencing instrument or in certain runs of a sequencing instrument. So if you do, let's say you have your control samples and you have your treatment samples and you run all your control samples on Tuesday and they all look beautiful, and then you run all your treatment samples on Thursday and there's a whole bunch of technical variation, then all of a sudden your stats return things that aren't real, that were caused by the instrument and not caused by actual biology. And this was sort of came to light as a much big, even though I said technical reproducibility is very good in RNA-seq. I still think that's true. I just think you can reduce it by randomly blocking your controls and treatments across any like different library preps or different instrument runs you're going to do. And this became very much in the focus of Twitter in particular. This was a huge Twitter storm a few years ago. There was this PNAS paper that came out, so a very good journal, comparing the transcriptional landscape between human and mouse tissues. And they said RNA-seq varied more, like the groupings were tended to be human and mouse and not tissue specific. And they're kind of tearing down mouse as a good model for human research. Well, this group, these two guys came along and said, we think that your entire statistical design and results are complete crap. And they published an entire paper doing nothing but calling out this PNAS paper for poor methods and poor statistical design and poor blocking. In very no uncertain terms, these guys did a terrible job, like maybe not career wrecking, but certainly very, very embarrassing. So you can go back and look up there's blogs and there's people yelling at each other in the comment section, and it got really, really crazy. But this is not who you want to be, right? Like, well, you don't want to be these guys in PNAS paper. You probably don't want to be these guys either, but you definitely don't want to be the PNAS paper author who gets called out in public for doing a bad job. So what do you do about this? Let's say you're going to run your samples on 7 different lanes or 7 different runs of an instrument. Instead of doing something like this, where you have each one of your samples is running in its own lane, and you could have technical variation, it's very easy to wash this out of your experiment by just running all of your samples on every lane. And this is something your sequencing course should be able to do pretty easily. So if you do have to do multiple lanes, just try to run all of your samples on all of your lanes. Yep. Within the same lane? So that's harder to control for, but if you're, well, yeah, so it's a glass slide, and your little RNA-seq, well, there'll be cDNA at that point, attached at different points, and then the camera's pointing up and seeing all the little points of light. But what happens is when you mix all of your samples together, they get randomly arrayed on that slide. So if a sector of the slide fails, it should fail equally for all your samples because they're random, right? Like once you barcode and put that all onto one lane, where they end up on the slide is random. So you can get bad sectors on the slide, but it shouldn't impact your results. You can wash all that data out because all those molecules are just floating around randomly once you mix them together. So that shouldn't actually be a problem, but that can happen. It just shouldn't, if you have added all of your libraries to the slide or to the lane, it should be fine. Yeah, there's other things that are harder to account for, like if you have to switch, if you run out of all the reagents in your library kit and you buy a new kit. It'd be nice if you could do like half your controls and treatments and then switch to the new kit and do the rest of your controls and treatments, instead of having like all of my controls are done with like the kit that was 2 months expired and all my treatments are done with like the brand new kit that's fresh, and maybe they've like changed some reagents. So if you can reduce your technical variation in any way, I would encourage you to do that. Again, I don't think that's the main source of the variation, but don't risk it. Any questions about that? Stats is fun. Read count, how to decide. So I already talked about this a little bit. This is from June 2011, but I think it's held up surprisingly well. The ENCODE consortium basically said, if you're comparing 2 mRNA samples for differential expression or more, 30 million paired end reads per sample is fine. If you want to discover novel elements, perform more precise quantifications, especially of very lowly expressed transcripts, they recommend 100 to 200 million paired end per sample. I think this is still pretty much what I see people do. Is 30 million is going to get you 10 to 30. I tend to err on the side of like you can go lower and you're going to get most genes, especially if that means you can afford biological reps. The 10 to 30 million paired in reads per sample is great. If you have a well-sequenced reference genome and you know all your gene models, you maybe don't even need paired in sequencing. You probably want to do like 75 base pair single in sequencing and save yourself some money there too.
When you say 30, what reads for count one?
Yeah, so like 30 million pairs. So you'd actually have 60 million reads. Unless you do single in which case you just have 30 million single ins. But no, like it would be 60, yeah, 60 million reads, yes. But if you know that you're looking at lowly expressed transcripts, like maybe you're working with a pathway that you know is just, it's there, but it's not highly expressed, you do need to go to much more depth. And I already showed you this. So there's another paper. I would claim that beyond, you know, like 10 to 30, somewhere in there is about where the literature falls per sample. If you have to choose between depth and replicates, always choose more replicates. Also look around at what's being published in your community. I feel like it kind of differs depending on if you're working with bacteria or plants or mouse, or, you know, your community is going to be different. And also looked at what resources you already have. If you have a well-assembled and annotated genome, you can save money by using single ins, shorter reads. If you are going to be doing your own de novo transcriptome assembly, you want longer reads, you want paired ins, you want to go ahead and pay more for that. Another new kind of cheaper, easier, fun protocol is the 3' RNA-seq, which I think is also called 3' tag-seq. And this is basically, instead of traditionally, let me see if I get my pointer, where you have all your mRNAs and you're fragmenting them and you're sequencing all parts of the transcript. You take your mRNAs, you fragment them, but then you only sequence off of the poly A tail. So you're only getting this one little region. You never sequence any further into the transcript. So this actually has a number of advantages. It requires fewer reads for the same statistical power, right? So in this case, we're able to quantify these genes using just a handful of reads versus having many, many reads that cover the whole gene. Easier library prep costs much less. So you could save over half your library prep cost. Single read sequencing is sufficient, so you don't need to do paired ins. That's also going to save you a lot of money. The big disadvantages, no transcript splicing info. So if you are interested in alternative splicing, you're not going to get that. You do have to use the poly A tail. So if your organism doesn't have a poly A tail, this won't work. And it's better for organisms with reference genomes. And here's a study that kind of said, you know, maybe we could use this for organisms where we're going in and we don't know, we need to do a de novo assembly, and we'll just de novo assemble the 3' end and we won't know what the rest of the gene looks like. But the conclusions of this paper from last year were basically that it's not worth it. Standard RNA-seq presents a better cost-benefit compromise if you don't have a reference genome. Whereas for model species, the 3' RNA-seq method it might more accurately detect differential expression. So in case you're pricing something like this out right now, something to think about. So I'm going to talk a little bit about the lab, which is going to be a standard protocol for differential gene expression, where, as I mentioned, there's many other things that you might want to use RNA-seq for, like genome annotation or catalog of genes, but that's just not what we're going to be doing. If that's something you're interested in, feel free to come talk to me and I'll tell you what software we use for that. Some of these might have changed, clearly, because Narola designed the lab, not me. But the beginning part looks surprisingly a lot like the DNA-seq that we already did. You still want to look at the quality of your data. You still want to do some trimming. Well, it's not necessarily true. Some people have quit trimming RNA if you're just mapping and quantifying. They say it's not worth it. If you're doing an assembly, you definitely still want to trim. If you're just mapping and quantifying, that is at this point considered largely optional. You want to map your reads back to the reference genome, but in this case, we are going to want to use something that is specifically for RNA. You want to use something that's splice aware, that will use your gene models, that will know, because your reads will be split across those introns, you need to use something that's specifically designed for RNA. At that point, just like DNA-seq, I do recommend that you go look at your RNA and see, is it lining up on the genes? Do my gene models make sense? Like, has everything I've done up to this point actually yielded something sensible? From there, you want to count the reads per gene. We use something called HT-seq for this in my lab. There's a number of different ways to do this right now that are all pretty interesting, but we'll demo HT-seq. Differential gene expression, I think we're going to do DE-seq2. And then there's all sorts of downstream stuff you can do, looking at like go-term enrichment. So if you get 10,000 or you get 3,000 differentially expressed genes, how do you know what they're doing? You don't really want to look at 3,000 genes one by one. So there are some ways you can kind of summarize, like what pathways are they involved in? Let's do a KEGG analysis. What types of physiological processes are they involved in? You can do a gene ontology analysis. And we like to use Bingo for gene ontologies in particular, and KEGG for pathways. And then way back at the beginning of the class, we learned about submitting to SRA, which is also something you'll be expected to do for RNA-seq data, not just DNA-seq data. If you don't have a reference genome, you're going to need to take a little side path through this, where after you definitely do your trimming. You may even want to do error correcting. Then you do your de novo assembly, and then you're going to be mapping your reads back to your assembled transcripts instead of back to a genome. At this point, you might also want to do some functional characterization of your brand new transcripts. At that point, you can kind of funnel back into the same pathway. A little bit about mapping to the reference. Mapping to RNA is a bit more complicated because you do have introns and alternative splicing. So you want to use mapping software designed for RNA-seq. You're going to want to provide the software with a file, with a file called a GFF3 file, so that it knows where the genes are located. So it knows like an intron starts here and an intron ends here. So I expect to see reads that stop here and then pick back up over here, like that intron is gone. The software is more accurate if it knows where the introns are. If you don't know your gene structures, but you have a reference genome, or you think there's new genes that haven't been annotated correctly, some RNA-seq mapping software packages will actually try to tell you where there are genes. You can go look. But the important thing for this lesson is that you have some general idea of what a GFF format file is, so that you can provide it to your mapping software. This is another one of those great stories, kind of like the FASTQ, where like the formats got kind of out of control and crazy, and now we're left with a bit of a mess. But GFF was the original file format, and it's just a way to represent genomic features on a sequence. For example, this is where a gene lives on a chromosome. It didn't really cover all the use cases that people needed, and eventually different groups were kind of like extending it in all different directions, and nobody's GFF files would work with anybody else's software. And it was really confusing and difficult, and nobody could figure out what GFF even was. So now we have GFF3, and this was the attempt of the community to say, okay, hold on, we got to start over and make a better, more tightly described format that will actually work for many use cases. It's an attempt to add and standardize the most common extensions to GFF, preserve backward compatibility to some of the GFFs that are still out there. And we already talked about backward compatibility with Python. And it's basically a 9-column tab delimited plain text file. So we've seen a lot of these. So this should be familiar. It's just another series of columns of stuff. So the first column is always going to be like your reference sequence. So like chromosome 1. The next, if you have a source for this annotation, so if you want to put down like a curator's name or a piece of software, or like there's some source that this is who told me this is where this gene lives. The next thing is a type, like what type of thing are you giving the location for? It could be a gene, it could be an mRNA, it could be a repetitive element, it could be a SNP. It's a location of anything. So you have some keyword to say, what is the thing that we're locating? The start position and the stop position. You can give it a score. So if it was computationally derived, you can say, oh, this had a score of 10, this one had a score of 100. You say the strand, so it's on the positive or negative strand. A phase, so if it is coding, you can say what phase it's in. And then you have the catch-all attributes line, where there's a list of feature attributes in the format tag equals value. The most common one is like ID equals, and then it tells you the name of this thing. So if it's a gene, it'll be the gene name. If it's a repetitive element, it'll be the repetitive element name. But there can be many, many different tag value pairs separated by semicolons. So the way that this format is actually quite clever is that it uses its parent as part of that last little bit of the description. And by having a parent tag, you can have a hierarchy of things that live on a chromosome. So you can locate the gene, and then you can locate the actual mRNA, and then you can locate each individual exon and each individual 5' UTR and each individual 3' UTR. So each of these gets its own line in the GFF file, but they're tied together by that parent equals. So you would specify my gene is from 1,000 to 2,000 bases. Then you say my mRNA, it actually starts at 1,050 bases and goes to 2,000 bases. This is mRNA has the parent of the gene. And you say one of my exons goes from 1,200 bases to 1,300 bases. Its parent is this mRNA. Then you can have the next exon, whose parent is the same mRNA. Then you can have your 5' UTR, and its parent is the same RNA. So you can comb through the file and you can recreate the entire gene structure with a series of lines. This also means, through this parent system, you can have alternative splicing specified in these files. So you could have two mRNAs that have different exons, but they both inherit the same, they're both children of the same gene. So this is how we keep track of all of the different isoforms that have been annotated in all our genomes. And when you do a head or a tail, this is sort of what it should look like. So like this is contig 123, example source, let's see, mRNA 1050 to 9,000 on the positive strand. This is the ID. But then you see each of these 5' UTRs, CDSs, and 3' UTR have the parent of this mRNA. So you know they all group together to make one gene annotation. This is actually when you're looking at a genome browser and it like draws the little boxes for the exons and the little like lines in between them, so you know they form a gene. All of that is coming from this format. Well, usually from this format. There's another one called GTF. We're not going to get into that. They're actually competing formats for the same type of data. We're going to use a GDFF in this class, and so that's how you specify where things live and how they make features and how your genome is annotated. All right, so that's all I got. We can move on to the lab. Are there any questions before we do that? Oh, that made 100% sense. That's exciting.
So.
You're like, no.
So this might be a really dumb question.
No.
Do you make the GFF?
If we,
programs out there that do
yes, we annotate the genome with software, Breaker, Maker, or whatever, they produce the GFF for us. And nobody ever sits down and hand types it, ever. And if you have a reference genome that lives in NCBI or lives somewhere, it will generally come with a GFF. You just have to be looking for that file extension. Like there will be some download you can go get and it will tell you where all the genes are. But no, nobody's sitting there and typing that because it would be crazy. Yeah. But it has to be formatted somehow for computers to read it, and that's how it's formatted. So yeah, Daniel.
Is there any platform where they cross-check the annotation?
So the platform that you do for manual annotation, the only one I know of is called Apollo. And it's a very nice visual website-based system. And you can align genes from other organisms and align your RNA evidence. And then you can actually drag and drop your exon boundaries and like your UTR boundaries. And like you can play with it in this very visual, intuitive way. And then you click save and it spits out the GFF3. So you don't have to type it. But you can sit there and like people do like manually curate whole pathways in some of these organisms to make sure they're perfect and they've really nailed like every codon and every isoform. There's automated methods and then there's these very manual methods. But that's how, if you wanted to alter a GFF, your best bet would be to try to find somebody with an Apollo setup and get you going on that. Yeah. Hand annotating GFFs, we've done it before, it's really hard. And so it's very tedious. Any other questions? It's just important for you to know that you're gonna need that to map your RNA-seq well. You don't have to write it, you don't have to produce it, you just need to be able to find it and know that that's what you've got. And I think it's good for you to understand when you run these software tools, why? Why am I giving my RNA-seq mapper a GFF file? So it knows where the boundaries are for your intron so it can map the reads more accurately. That's sort of the take-home message here. Not that you guys need to write these files necessarily. You just need to be able to find them and put them into the software and know why you're putting them into the software. Any other questions? No?